Hello, friends. We are back. Last time we talked about basically primary structure, the nature of the peptide bond, and some constrictions on the angles. We're going to go a little bit deeper into that this time, but we're going to spend this session talking all about secondary structure. And if you remember from general biology, secondary structure is essentially this combination of these three structures: alpha helices, beta sheets, and the beta term. In GBio, it's generally enough to know that these structures exist, but we're going to talk about why they exist and why they predominate in the protein structure. Next session, we'll talk about tertiary and quaternary structure and some of the motifs that come up. But now we'll talk about this. So the alpha helix is, as it sounds, it is a helical structure or a spiral that turns around itself. It is stabilized by hydrogen bonding within the peptide backbone, between peptides, and these hydrogen bonds occur between nearby residues. And we'll see that hydrogen bonds occur with residues that are 4 amino acids away. Whereas for the beta sheet, it's also stabilized by hydrogen bonding within the peptide bond, but those bonds can be formed by residues that are hundreds of amino acids away. And we'll see why that is in a second. So the other thing, kind of things that aren't connected by the alpha helix or random sheet, are called random coil, and they are kind of like they sound. They don't really have a distinct shape or structure. So remember when we talked about the peptide bond, we said that because of delocalization of the electrons within that bond, you had a forced planar perspective. And so the peptide bond forces a planarity. You don't have free rotation around this bond here. Let me just find another one. Um, Around this bond. It is forced in a particular position. You still have free rotation around the bond between the nitrogen and the alpha carbon. That is called phi. And between the alpha carbon and the carbonyl carbon, that is called psi. So you have relatively free rotation around phi and psi, but you don't around omega, which is this bond between the carbonyl group, and the nitrogen of the amino group. Now, that phi and psi, you do not have full 360-degree rotation around those. There are going to be some constrictions to the rotation around phi and psi, and we'll look at what those are in a second. Most of those have to do with steric hindrance, or these functional groups bouncing and bumping into each other and causing issues or constraints onto the structure. Some have to do with the ability to maximize the amount of hydrogen bonding that occurred between that dipole within the peptide bond. We have something called a Rama plot, a Ramachandran plot, that allows for the allowable configurations of phi and psi. And it looks something like this. And these are a little bit difficult to kind of break down, but essentially what we have is 360 degrees of rotation, right? For those two angles, phi and psi. Remember, phi is the nitrogen carbon, and psi is the alpha carbon carbon, right? And so there, this shows each of these dots represent the phi and psi of one particular amino acid residue in one protein, right? And so each of these dots, let's say this protein, I'm not good with spatial numbers here, but let's say this is, I don't know, 250 amino acids or something like that. Each of these dots represent what the phi and psi is for one particular one within that particular protein chain of a polypeptide. And so if you're looking, and I'm just making this up here, but let's say this is an alanine, They'd say this is the observed angle of phi and psi for that particular one. Let's say this is a cysteine, observed angle of phi and psi, and so forth. And so if you just take a protein as it exists in its native conformation and plot each angle of phi and psi, you get something that looks like this. And there should be something that comes out, right? There are angles in phi and psi that are poorly represented within this plot. So for whatever reason, you don't see a lot of angles of phi and psi that correspond with this configuration. Let's say negative 20 on psi and 120 on phi, right? Here's another area where there's a lack. There's a little bit over here, but for the most part, this quadrant is missing out as well. You note and see that there are some areas on this Rama plot where there's a lot of angles of phi and psi. So up here at about 180 in each way, right? You see pretty good representation. Here at about 60 for psi and maybe the same for phi, you see a lot of representation, right? So what this tells us is that there's some uh, configurations or angles of phi and psi that tend to be favored in a polypeptide, right? And so the question is, what secondary structures does this correspond with, and why it is that those tend to be favored in polypeptides? Here's another look at this. So this is, in this particular case, this is a look at dozens or maybe hundreds of proteins, and you see a similar result to up here, right? You see favored configuration here, a favored configuration here, and another maybe favorite configuration here. Not much over here, very little over here and over here, right? So that's looking at dozens. And if we were to pull this apart and look at particular amino acid residues, we will see that some amino acids have a little bit higher degrees of flexibility. Note for glycine, um, here you have a little bit more that's able to happen, right, relative to uh, the every amino acid configuration. You see more representation up here, much more down here, right, and then more that's over here in this area. On the flip side, something like proline, you actually see fewer degrees of flexibility, right? And so for proline, you see some here, and you see some here, but no, it's completely absent all the way over here, right? And so my now 10th favorite question in biochemistry is, why is it that glycine has what appears to be more degrees of freedom, whereas proline seems to have much less, right? And so, what is it about these two amino acids that have such a profound difference in the number of possible Raman plots that are available? So, when we're looking at this Rama plot, I said that there are some areas that are preferred, and it turns out that this area, which we see right here, corresponds to the alphas, the angles of phi and psi that correspond with a right-handed alpha helix. Here, this area up here, which we saw here, corresponds to beta pleated sheets, either anti-parallel or parallel beta pleated sheets. Right? And this area here that we see right there, that corresponds with a left-handed alpha helix, right? So one of the answers to the question as to why the secondary structures are maintained is that the these particular secondary structures of an alpha helix, a beta sheet, correspond with allowable configurations of phi and psi that minimize the steric hindrance of side chains and other things crashing into each other, right? So one of it is just that this gives space for the side groups, or for the side chains to spread out and be a little bit comfortable. Uh, the other reason is that these configurations maximize the nature of, or the amount of hydrogen bonding within the peptide backbone of the polypeptide. So I think I'll stop here to try and keep it under 10 minutes, and then we'll talk about the particulars of the alpha helixes and beta sheets in the next session.